Okay. So next week, if you want to meet me, forget it. It's Thanksgiving, you know. That's how it is. Actually, I'll be here during the week, but from Thursday on. I guess I could invite you for Thanksgiving dinner, but we'll see. No test today, right? We're going to have it on December 2nd. What I might do is post what I cover in my other 100 class. I might post a lecture because we got some stuff to cover, and I might ask you just to look at that. But in the meantime, we can start with this. We already saw this, actually. Properties of cancer cells in terms of immunosuppression. So, I don't know this for sure, but I'm pretty sure. I think all our cells do this. I think there's a very delicate interplay between our immune system, which we'll be learning about, probably when we get back, and then our own cells, so that they're not targeted by our immune system. So sometimes our cells are targeted by our immune system, and those are the source of autoimmune diseases like multiple sclerosis. There's a slew of them, where the body attacks its own cells. Okay, so what the cells do, our normal cells do, I'm pretty sure, is that they release chemicals and they utilize cells to damp down the immune system in their immediate area so that they're not attacked and they can function properly. Well, cancer does the same thing. It generates these immunosuppressive microenvironments, like around the cells, and it then triggers a cascade of a whole bunch of different things, including immunosuppressive molecules. You can see down here. Okay, so and also immunosuppressive cells. There are these signal inhibitors that affect cancer. And then they do a whole bunch of stuff. They let dendritic cells, which are kind of the basis of part of our immune system, in terms of recognizing foreign things. So cancer has a way of hiding from our immune system. Now, of course, we've got tumor suppressor cells and all that for our immune system, but it still gets away a lot of times with it. So, and then talked about this as well. So immunotherapy, this is the latest advance in cancer treatment, where you harness the immune system to fight the cancer. And there's a bunch of different techniques, including checkpoint inhibitors, monoclonal antibodies, vaccines, adaptive. You don't have to know all these, right? But the thing is, the first thing that cancer, what needs to be done is to disable the cancer's immunosuppressive system on the immune environment. In other words, to stop cancer from shutting off the immune system so that it can then go on and attack cancer. So, and that's why it really wasn't, people thought of, trying to use the immune system to attack cancer before, but they didn't know enough to realize what was actually going on. So now they have a better idea of the kinds of things that they can do to actually get the immune system to attack cancer, and only the cancer, and nothing else. So we, let's see. Oh, that's right. Okay. Um, I put up a short video on meiosis that goes into a bit greater depth of what happens during meiosis. There's also a video on the Moodle page that gives it actually carries you through what happens during meiosis. But there's a few things that I want to point out. Now, sexual reproduction, there are costs and benefits. The costs are that if we remember about evolution, the more copies of your genes you pass on to future generations, the higher your fitness. Okay? Well, that means since you're 100% related to yourself, and you make a clone that's identical to you, and not compared to with sexual reproduction where 0.5 related to our parents and 0.5 related to our siblings, there's a real benefit to just reproducing asexually. So the question then becomes, well, why is sex so common? And because it has to overcome like a twofold cost. First of all, if you have 10 cells, let's say, or organisms, whatever they are, and they can leave 10 copies of themselves in terms of reproduction, and then you compare it with 10 individuals who use sexual reproduction, they can only leave 5 of themselves because they need a male and a female to produce one organism. And that organism is only related by 0.5. So why is there sexual reproduction at all? Well, obviously, if you're a clone and things don't change, it's pretty good. Your life is pretty good. you don't really need to have a great diverse genetic diversity because you're kind of already adapted to the local environment. But if you're in an environment that's heterogeneous, that means spatially there's different parts where certain alleles are better than others, different time frames, so temporal variation, then having sex actually helps. First of all, you can create a lot more genetic diversity with sex and maintain it, and then just by happenstance you might end up with offspring that are, let's say, already, they have a mutation that makes them resistant to some new environmental factor, whether it's a pathogen or a better way to get food, all sorts of different things. So that's one thing that's going on as far as sex. Just this kind of like, the more diversity you have, the more chances, just by random luck, that you're going to actually have some advantage in different sorts of environments. Now, so that's part of the advantage. Now, the immune system itself generates a lot of diversity, and I'm kind of going to go through exactly what happens. As you've remembered, we're diploid, so we get one chromosome from mom and one chromosome from dad. Okay. Now that's not entirely true. During prophase, and we can take a look at this up here. So during prophase of meiosis, okay, we've just come out of G2. One of the first things that happens is homologous chromosomes. That means one from mom, one from dad. They line up together. And they actually do something called crossing over. And so, this is what it looks like. And so now you have, over here, now this is part of the recombinant chromosome. In other words, it's got a chromosome that's mostly from, let's say, dad, but it's also got a small piece from some other chromosome, mom. Okay? And then, of course, the non-recombinant chromosome is this down here. So, let's see, non-recombinant, and then here we've got recombinant. Okay? This crossing over takes genes from one chromosome and switches it onto another. And it's called genetic recombination. Yeah.
Whenever I look at these kind of photos, they always show the exact same part that is crossed over.
Yeah.
Can it be like any part of the chromosome, or is it only that little specific area?
They have to line up. Okay. So in this case, if you remember about meiosis, there's actually these tetrads that form because you've got double the number of chromosomes because you've replicated all your chromosomes. And then these are going to actually, each one of these is going to turn into a part of a daughter cell with just one of the chromosomes. Okay? So it could be, and that's during meiosis too. And so the crossing over, it is fairly random, but it has to do with chromosomes that are kind of lined up together. Okay? Okay. So that's the first thing that happens. It actually doesn't happen, it's during prophase, not metaphase. Metaphase is something else. So, and then, here we are. Now, there's also something called independent assortment. If we look more closely at this, okay, and this, here we have on the left 2 homologous red chromosomes and 2 homologous blue chromosomes. And then here on top we have a homologous red, and then we have this recombinant one. So instead of, it's kind of like the opposite of this one. So it's, the way it happens is kind of random in terms of which way these chromosome pairs are going to go to opposite sides of the cell during anaphase 1. And then, let's take a look. The same thing happens during metaphase 2. Now, of course, we've got 2 cells that, because things have been reduced to metaphase 2, and each one, there are a total of 4 total chromosomes. During telophase 2, anaphase and telophase 2, one of these chromosomes, let's say, that one goes over there, and this one on the bottom goes over there. And remember, these are now haploid gametes. In other words, sperm and eggs. So each gamete only has 1 chromosome. In terms of humans, we have 23 pairs of chromosomes. They have 1 pair. Whereas in diploid organisms like ourselves, we have 2 pairs, 1 from mom and 1 from dad, plus all the crossing over and everything. Okay? So you get this very diverse group of what are called haploid gametes that can have all sorts of different parts of the genetics based on recombination through crossing over and also independent assortment twice. That's why each human being, salute, is actually kind of unique. That's why you don't have identical individuals unless people are actually identical twins. Okay? And that's why sex gives all this genetic diversity. On its own, if things didn't change at all, it wouldn't matter. It'd be better to just be clonal. But since we're in a very kind of dynamic world in terms of environments, sex actually is advantageous. So, this says what I just went over. You get this genetic diversity from genetic recombination by crossing over, independent assortment. So we have 23 pairs of chromosomes. So we take 2 and raise it to 23rd power, and we get 8 million types of gametes. Now, there's also another thing going on. Random fertilization. Which sperm actually manages to get to the egg first and fertilize it is random. And so you can end up with something on the order of 64 trillion genetically different offspring that your parents could potentially produce. So a lot of genetic diversity. Okay. Let's see here. I don't want that. And here's dizygotic twins, of course, same thing. Okay. Now, here's a question. This is a little roundworm called C. elegans. And production of sperm, which is stored, so it produces sperm for itself, and it stores those sperm. It can then switch later to producing oocytes. That means eggs. Okay? In other words, it's one sex in the beginning, and it starts acting like the other sex later. So this is called a hermaphrodite. In this case it's actual sequential hermaphrodite, but just a hermaphrodite. In other words, an individual with fully functional, able to produce sperm and eggs. And then it can actually self itself, use the sperm that it first made and fertilize the eggs that it's making, and then produce diploid organisms like itself. Okay. So, is this sex? It is, of course. It goes through the same whole business. You have a diploid adult. They release, they create gametes during meiosis. The haploid form is in the gametes. Two chromosomes in each gamete. They then come together and fertilize and recreate the diploid. So, sex is not exactly how, or as simple as what you've been brought up to believe. In fact, sex is a lot more interesting in a lot of other animals and plants and all sorts of different things. Now, just so you know, in terms of sexual reproduction, well, first, asexual reproduction. The individual, let's say it's a single-celled organism, it makes a direct copy of itself, it's a clone of itself. Okay? There's no meiosis going on, none of that stuff. You make an identical clone of yourself. No gametes get together, nothing. But with sex, you actually produce gametes, and then you fertilize to get a zygote, and the zygote then starts dividing for mitosis and differentiating into different cell types until finally, in humans, you get a baby, and that baby actually keeps developing until probably their very early 20s. Okay? So there's still not things haven't been settled, especially you can imagine with little babies, they can hardly do anything for themselves. So there's all this kind of stuff going on, dividing and differentiating and all that. So, okay, your somatic cells have 2 of each chromosome. They are what kind of cell? Haploid or diploid? What do you think?
Haploid.
No. It's okay, fine. So, if we're somatic cells, that's most of our cells are somatic cells, right? They are diploid. They have 2 chromosomes for each chromosome, or a homologous pair of chromosomes, one from mom, one from dad. Okay? The only thing that is haploid is during the gametes. Okay? So that's the only thing that's haploid. In other words, the adult has to undergo meiosis, produce gametes, and then those gametes have to reach each other during sex, and the sperm fertilizes the egg. So the gametes each have a single chromosome. Could be from mom and dad plus all the crossing over and everything. And then when they fuse during fertilization, you end up with a zygote and a diploid organism. Okay? And then that zygote, that single fertilized cell, then undergoes division after division after division, and then later differentiation into different tissue types, all sorts of different things. Okay, so if we put that here. Okay, so what are these pairs of chromosomes called? Starts with an H. Anybody? Yeah, homologous. Okay. Specialized form of cell division that occurs in your germline cells. Meiosis or mitosis? Okay. Gametes have one of each chromosome, so they are haploid. Okay, so this is this kind of instruction manual, but it's not that simple, of course, because the environment is involved in gene expression. Okay. This is our hemoglobin gene, and this is the protein that carries oxygen in our body. It's like any other protein. It's made up of amino acids. So you can see CTG that codes for leucine. Let's see, lysine, serine, all that kind of stuff. Now, one of the things that make up hemoglobin is iron molecule that attaches to oxygen very easily, and that's why iron is selection eventually favored for iron. But there are some animals that actually have green blood, and they have a copper-based blood cells for doing the same thing. But hemoglobin is iron-based, and it's more efficient. Okay, so this is a normal hemoglobin, right? And we'll call that HbA. Okay, now you can also get an abnormal hemoglobin gene or hemoglobin molecule, and if we actually look right here, we see that the GAG has now, the A has been changed to a T, and we go from glucine to, I think valine is what it's called. Okay, so it's just a one letter in the genetic code, a single mutation that then changes the amino acid. And this is the complete hemoglobin gene in terms of the coding of it, and it's on chromosome 11. So, okay, now, this is what the protein looks like in terms of normal and in terms of abnormal clumping. And since our red blood cells are made of hemoglobin, we have a normal cell and an abnormal cell. And so this abnormal cell, it has two copies of the mutant gene HBS, and it produces a sickle shape. It is not very good at carrying oxygen, so that's why the person is anemic. Plus, in lower oxygen environments, it gets more sickly, and it then clumps in the, not the arteries, but the smallest arteries, or the smallest carrying oxygen-enriched blood, and causes all sorts of problems. And it's actually, in this case, when it's HBS, it's actually lethal. They don't produce, there are people that live with it, of course, with hemoglobin problems like this, but they usually don't produce any offspring. Their fitness is really low. Okay. Now, we've got some different words. So if we got the two alleles, the normal is HBA and the abnormal is HBS, we have a heterozygote. You could do the same thing. Let's say allele A and allele B. Doesn't matter. Whatever the allele is, if it's in two different alleles, you've got the heterozygote. Now each person, since we're diploid, we each have an allele from mom, an allele from dad. Now, you can also have, let's say for mom and dad, you produce two normal hemoglobin alleles. And they give them to you, and you end up with normal hemoglobin. Okay? And then, in this case, this is homozygous, recessive though, and this is the one that causes sickle cell anemia. Okay, so let's take a look at this. The genotype, homozygous normal, HBA, HBA. The phenotype is normal red blood cells. HBS on the bottom is sickle cell disease. And then this one in the middle, the heterozygote, HBA, HBS, is resistant to malaria. It also shows what's called incomplete dominance and codominance. We'll get to that. But it has the highest fitness in an environment where malaria is endemic. Okay? So someone with the heterozygote is resistant, the parasite can't get in to their blood cells because their blood cells actually have a little bit of sickling. Okay? Now, I just want you to know something. If you take the person with, let's say, the heterozygote and compare them to the homozygote in an area where there's no mosquitoes and malaria, then that genotype is actually has lower fitness than the homozygote HA. Okay? That means it depending on the environment, whether an allele and a trait is actually adaptive or not. In one environment, the heterozygote is adaptive compared to the normal allele because the normal blood cells will get infected with malaria and a person will get very sick and maybe die, especially in childhood. But you take that person out, and heterozygotes then have lower fitness because they actually pay a cost in terms of the fact that some of their cells are actually somewhat sickle-shaped. So, I've mentioned these things again. So we have a genotype over here, right? And you end up, there's two alleles. you have one allele from your mom, one from your dad. It can be the same allele or it could be different. And then you have the phenotype, which is the expression of the genes in terms of the proteins made. And you end up with these different phenotypes. In terms of which has the higher fitness between these two and this one up here, it's determined entirely by the environment in which they live. Normally, normal blood cells, with there's no malaria around, has the highest fitness and carry the most oxygen. There's never any anemia whatsoever. But when there's malaria that's endemic and likely to be, someone is likely to be infected, then the middle phenotype has the advantage. Now, heterozygotes right? They're carriers of the HBS allele. Okay? Having this allele makes the individual resistant to malaria, but they can also pass it on to their offspring. Now you might say, well, that's good, except that if you get two of those sickling alleles, you end up with a genotype of hemoglobin that's HBS, and you end up with sickle cell anemia, which has very, very low fitness. Okay. So I want to go over a little bit. This is kind of, we'll take a look. Okay. Oops. Okay. So that first generation, we'll say these are flowers, and we have an adult with white flowers, and we have an adult with red flowers. Okay. Now we're looking at dominance here. If you actually look at, let's say, this parent has two white alleles, and this one has two red alleles, they produce offspring that are all red, but as you can see, they're heterozygous. They have a red allele and a white allele. So all these guys, these offspring, are heterozygotes, okay? All of them. Now, you go to the third generation, and you start crossing these heterozygotes. What do you get? Okay, so if you have one copy or two copies of the red allele, and the flower is red, that is a dominant trait. Okay, so here, let's see, actually. Okay, now, you can also get something. So here we are with our generations. We have the same white and red. And they each produce identical phenotype, and that is they're all pink. Okay? And they're heterozygotes. That means they have a red and a white allele. Or they have a red and a red allele. Okay, so the red gives you red, the red-white gives you pink, and the white-white gives you white. So this is called incomplete dominance. And then finally, So again on the top, this is dominant and recessive, but you can actually have something called oh, and here's incomplete dominance, right? Pink. You can also have something called codominant, where parts of the flower are red, that means RR, and parts of the flower are white, WW, but they're actually heterozygotes. But they, instead of turning pink flowers like intermediate, like mixing paint, each of the pigments are pure, but they're found on the same flower. You also see this in cattle, where you have different color splotches. Let's say it's brown overall, but you also have white areas, you also have black areas. That's another example of codominance. Okay, so we'll say male and female. The female has a red, and she also has a white, and the male also has a white and a red. Okay, and when you have two R's, that's the dominant. It's homozygous. You got the same copy of the allele, and it's red. When you have you go across from this R to this W, you also end up with RW, and it's also red. And here it's WR, same, but these are two heterozygotes. And then you also end up having the WW. Okay? So only the heterozygote expresses the recessive allele. All the other ones, whether they're homozygous dominant of red or heterozygote red and white, they still show the red phenotype. So here's this is all in words. If A1, there's two alleles. A1 is a dominant allele, and the other allele is A2. Okay, A1 and A2. If A1 is dominant to allele A2, then the dominant phenotype is expressed in both the homozygote and the heterozygote. In other words, A1 is dominant. Okay, so there's a dominant phenotype. And A1A2, which is the heterozygote, this one, it's still going to show the dominant phenotype. Let's say red flowers. A phenotype that is only seen as a homozygote, in other words, the same allele, but it's only expressed as that, as the same allele, let's say in white flowers. To get a white flower, you need two W's, two white flower alleles. So we call that recessive. A recessive allele is not associated with disease. It's only about expression patterns. We have a whole bunch of traits that are the result of dominance, codominance, incomplete dominance, all sorts of different things. Okay? So having recessive doesn't mean that you have some sort of a disease, though it can. Also, a dominant allele has nothing to do with the frequency of that allele in the population. So you can have a dominant trait that's very, very rare in the population, let's say, a disease called Huntington's disease. It's a dominant trait. You have one copy, you get the disease, and then you die a horrible death. It's a very rare condition, but it's still expressed in a dominant way. It only requires one copy of the Huntington's allele versus the normal allele. So the heterozygote will show it. I'm not sure about this. It depends, but you're probably never going to find a homozygote. In other words, two different people with Huntington's who breed. Well, first of all, Huntington's unfortunately doesn't show up until later in life, around 40 or 50. By that time, you've had offspring, and that's why it's still in the population. I don't know if you had the homozygous Huntington's allele, whatever it is, Huntington, whether that fetus would have even been viable or not. So I'm not sure. So some examples of dominant traits, freckles. Anybody can roll their tongue like that in here? No? Okay. Widow's peak, dimples, detached earlobes. I think that's what I have, yeah. Now, recessive traits where you need two copies of an allele. Now remember, these alleles, all they're doing is producing proteins, not traits. The proteins are what makes the traits. Cleft chin, albinism, that means you need two copies of the recessive allele, hitchhiker's thumb, attached earlobes, and it only shows recessive when you have two copies. Okay, so if you are heterozygous at a loci, let's say for the hemoglobin, and you have HbA, which is normal, HbS, which is sickle, then half your gametes will have HbA and half will be HbS. If you have the same allele as a homozygote, you'll have HbA or HbS. So remember, meiosis, the basic unit of meiosis are gametes. They're living things. And depending on whether the alleles are heterozygous or not, you can end up with two different kinds of gametes. Now, same thing for female. You end up with a little bit different, though. But you end up with eggs that can be either way. Okay. Remember this from before in terms of predicting. Well, we're going to do something similar now. We're going to predict. Okay. Hold on. Okay, so if you have two HbS carriers, let's do this. Okay, we've got male up here, W down here. We've got HbA. Okay, so we've got HbA, and this is HB, we'll just say HS. And the same thing, let's do it in red, HS, H, S, H, A. Okay, in the first square, you start, now what you do is, to figure this out, each individual produces two gametes, okay? They're diploid, so they have two different alleles. They can have two different alleles. But in terms of the gametes are going to be one or the other. Okay? So, in case of the woman, right here you have the normal type, same for dad. So in this place right here, you have H, A, H, A. Okay? Now, over here, you have the other allele, but down here you still have the normal. So you have H, A, and H, S. Okay, that's a heterozygote. Same thing down here now. These are both heterozygote people. She has one copy of the HS. And then there's an HA from the male up here. H, A. And then HS, HS. Oh, stop it. Okay, so these all add up to 1. So you have 25% of the people have normal hemoglobin, 25 people have sickle cell hemoglobin, and then these in the middle, who are carriers, these are the ones that are resistant to malaria, and they actually have the highest fitness when you're in a place where there's lots of malaria. Okay? And when you get that sort of a situation where the highest fitness is actually the heterozygote, that's called heterozygote superiority. And it's on here later. So here it is all. Okay. So this is sickle cell expression patterns again. In an area where there's malaria, you have HbA, HbA. That gives you normal red blood cells, but also being susceptible to the organism that actually causes malaria. And so you can actually see that here. Okay? And these ones have the parasite already in them. Okay. Now, okay, sickle cell, these HbA, HbS in the malaria area, malaria present, they're called sickle cell trait, not sickle cell anemia. They're malaria resistant. They actually have both types of blood cells. In other words, those that are HbS, HbS, sickle cell shaped, and HbA, HbS, which gives you the heterozygote, which is resistant. So that's why you can see this is actually a codominant trait. Okay? It expresses both phenotypes in the pure form. You have these sickle cell from having copy of HBs, HBS, but you also end up having relatively normal red blood cells. The thing is, they're not susceptible to malaria. Also, this disease shows incomplete dominance because it can carry oxygen as only about half as well as the normal type HbA. Okay? So its oxygen carrying capacity shows incomplete dominance. Now, we look at normal red blood cells, HbA, in a normal environment. They have the highest fitness. And you also, and they're dominant in a sense, they're pretty much, well, not quite dominant, actually. Let's get rid of that. They actually have the highest fitness. And then sickle cell anemia. Well, this is bad in malaria-infested environments or normal. Because when you have it, it means all your cells are sickling and you're anemic. And then they're gumming up the capillaries, and so all these organ failures go on. So, okay? Now, there's a whole bunch of genetic diseases. They're attributable to a defect in one gene. Okay? There are some that are the result of many genes all working together, and depending on which alleles of which genes are being expressed might also have something to do with a genetic tendency, let's say for cancer or something like that. Okay, so, but in this case we're looking at a single gene and two alleles. And with sickle cell, the recessive type is where the lethality is, and then depending on the environment, whether it's normal or heterozygote, you end up with HbA, HbS, let's say. And that's same thing with the cystic fibrosis. That's another one that's a recessive. It's usually found in Northern Europeans. It mucks up what is it? It produces very thick mucus, and so a lot of the people that have it have real lung problems, and they usually have lower fitness, but that looks like there's a treatment on the area. And then Huntington's disease, it's all you need is one allele, and it expresses the dominant phenotype. That means you end up with Huntington's disease. Okay. So, okay. The cystic fibrosis protein, normally it lets these chloride ions go through. A mutant one doesn't let as many chloride ions go through. So the recessive condition, how many of these alleles are needed for a person to be a carrier? Only one. How many are needed for a person to have cystic fibrosis 2? Okay, so, where is that? It is thought that the heterozygote might actually be advantageous in areas where, what kind of infectious disease is that? Oh, typhoid fever. Okay, So here we are again. If you have two carriers like it was before, a normal, you'll have normal. Here you'll have mutant and normal, and mutant and normal, and that's a heterozygote. I'm not sure if heterozygotes actually have less fitness in a non-typhoid fever environment. And then if you have two of the mutants, you have cystic fibrosis, okay, which is the lowest fitness. So remember, we'll say this is dominant and recessive. All these three express the same phenotype, and then the recessive one is the one you have cystic fibrosis. So this is a little different. If a man with CF marries an unaffected woman whose mother had cystic fibrosis, what is the probability that the child will have the disease? Okay, so here's the man. He's got one cystic fibrosis mutant allele, and he's got one normal. Okay? Now, the woman, we don't know, right? Is she homozygous or heterozygous? Well, remember, her mother had it, and to have it, you need two of the alleles. So the chances that she has one allele as a carrier is 50%. Okay? So, here we have two mutants. That means cystic fibrosis. Here's the heterozygote. Here's also the heterozygote. Oh, sorry, no, I blew it. Hold on. Okay, so here we are again. Okay, so we've got a woman who's a carrier, heterozygote, one, she carries a mutant allele and then a normal allele. And then we have a man who has two mutants. Okay, so the woman with the heterozygote, the mutant allele, these are both mutants. Okay? That means they have cystic fibrosis. Here, she also has a normal copy, right? And so she produces heterozygotes. In other words, they're carriers, but they don't have the disease. And that's also 50%. So it changes depending on, in terms of dominance and recessive, what you start out with. And you're going to have to do problems like these for the exam. Huntington's disease is a neurological condition. Anybody know who Woody Guthrie is? Okay. He wrote the song, "This Land Is Your Land, This Land Is My Land." Okay. One of the great American songwriters. It turns out his family had Huntington's disease. And he inherited one copy of the allele. And right around 40 years old, he started showing symptoms, which had like ataxia, you know, kind of spastic stuff. Also cognitive and behavioral effects as well. Okay? Now, because it shows up later in life, now normally if it showed up right away, selection would wipe it out, right? It'd have very low fitness. But since individuals can actually still breed before it actually shows up, it doesn't go away. There's maybe a selection against it, but it doesn't get removed, really. because you can still produce offspring. And so the kids don't know whether they're going to show up with the disease or not. Guthrie only had a number of kids, but the one you probably might have heard of was his son Arlo, who's another musician. Luckily for him, he was born with not the mutant allele. So he's in his 70s, 60s or 70s now. He's still alive. But it's a really hideous disease, and obviously you have a 50% chance of getting it. All you need is one copy. So here's Huntington's again. The large allele is abnormal, so the man has it. The woman is normal, and the man has a normal. So here we have here we have normal, abnormal, so again we have with a dominant trait, but this is because the dad basically has one copy and the mom is normal. You have 50% chance of having offspring with a disease, and 50% normal. Okay. Family pedigrees. Now, sometimes you see a situation where the trait skips a generation. In fact, we had that with dominant phenotypes. If you start out with a single, here, I'll do this. Okay. We have reds on top, or R's. And then we have whites. If R is dominant, all you need is one copy, so you have RR, 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 or sorry, WR, 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 right? That means you can have 4 heterozygotes. Since it's dominant, they all share the same phenotype as the father. Okay? It's not, and so they'll all be red. It's only when those individuals, in terms of the second generation, either carried the allele or not, that they then might show the other color, let's say, the white, in their offspring. So, in other words, these are all going to be heterozygotes, and then you're going to finally have some homozygotes that actually express the other phenotype. So, if a trait skips a generation, it's always a recessive trait. Okay, here. Let's see. Let's take a break until 12:30. Okay. Now, during meiosis, things can go wrong. And it's called, one of them is the most common one, is called non-disjunction, where you end up like here. This is a triploid at this spot instead of diploid. Okay? Now, and then you can also have a system where you get up only one, so it's haploid. Okay, so a lot of these mutations definitely have effects. Most of them are probably lethal, but some of them aren't. But they certainly produce certain phenotypes. So you probably are all heard of Down syndrome. And on chromosome 21, they get 3 copies of the 21 chromosome. And Down syndrome has a kind of a phenotype kind of like this face, and they have a whole bunch of other traits that are similar to them, and involves obviously many genes are on that chromosome as well. So, of course, humans have 23 pairs of homologous chromosomes. Good, we know that. Now, 22 pairs are somatic cells or autosomes, right? They're not involved in meiosis. They're only involved in mitosis, where they occasionally replicate. There's also a pair of chromosomes that are not actually homologous. So there's 6 chromosomes. The female has, looks like that up there, an X. A female has 2 X's. Okay. Now, and then the male has an X and a Y. Y is a lot smaller. Okay. So there's some stuff going on in terms of heritability with the X chromosome and the Y chromosome that is different from what the autosomes go through. So we've just been covering autosomal traits, okay, whether they're diseases or not. So what determines person's sex, the X chromosome or the Y chromosome? Yeah. So there's a certain gene that controls the production of testosterone and other sex hormones. And without the Y, it's only found on the Y chromosome. And otherwise, it's going to turn automatically into a female. Okay. Of course, there are hermaphrodite people who are hermaphrodites too. They're just getting their equality about now. So.
Wait, but for them, what do they have in their genomes?
Let's see. They have different, let's see, I'm not sure. Well, here might be the answer. Okay. So, you look at these sex chromosomes. Normal for a female is XX, and it's normal. And for a male, normal is XY. Okay? If you have an X, only an X and an O, which is kind of a placeholder, you'll end up with something called Turner syndrome. And if you end up with an XXY, you'll end up with a kind of a male genotype, but it'll be called Klinefelter. And then, let's say you end up with triple X's or double Y's, X double Y. They're all, one of these might actually be hermaphrodites, I'm not sure. Not exact. Now, sometimes hermaphrodites, it's not because of the genetics, it's actually because of the hormones that are present. Okay, now, you would expect the hormones to be under the genetics, but sometimes if if not enough testosterone is being produced or there's too much estrogen, it's going to feminize an individual who would normally express maleness. Okay? But you don't have to know that. So this is the SRY gene that triggers male development. It produces a specific protein. So obviously, the only one here who has a Y chromosome is the dad. So if he donates an X, then you end up having a girl, because you have females are XX. And then if he donates a Y, then it's a male, it's a boy. Okay. So there are these genes that are sex-linked. They can either be on the X chromosome or the Y chromosome. And they almost always show up in terms of male children. Okay, and we'll see why. So Y, males get their Y from father. Yeah. And then their X from their mother. So why do some genetic conditions affect males more often than females? Well, females are the homogametic sex. They have two X chromosomes. So if one of those X chromosomes, there's a gene that's mutation that doesn't produce the correct protein, there's often a rescue effect. Okay? So they still produce the protein from the other instructions from the other X. With the heterogametic sex, okay. 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 So if there's a problem on the X chromosome with a male, he only has one X. That means that he's got a problem. There's no rescue effect whatsoever. Now, there isn't always a rescue effect. You can have some situations where, the person is making half the amount of protein they need and it's not enough. But in general, because the males are heterogametic sex, they have only one X and therefore one Y. They're the ones who are the ones that are going to express phenotypes that are, you could say, disease phenotypes. Okay? Not all animals or plants or anything else are the heterogametic sex is males. In birds, it's the females that are the heterogametic sex. So it's just evolutionary history. Okay. So, red-green color blindness is caused by an X chromosome with a mutation in it. Now, women could get red-green color blindness if they inherited a faulty X from their mother and a faulty X from their father, who had color blindness. And the other one was a carrier. And then you could have female offspring that had color blindness. But in general, if you end up with a faulty X and it goes to the male, he's going to have it because he's got no rescue effect whatsoever. Okay? So if a colorblind man marries a woman who is a carrier for the recessive allele, what's the predicted outcome for their children? Oh. Okay. Okay. So, and this is the woman, the man. Here we have at the top, left corner, we have an X from the man, or from the woman, and we have another X that's a mutant from the man. Okay, that's a carrier. Here we have a normal X and a Y. Oops. Y. And here we have, oh, sorry, that's the woman, that's the mutant X. Okay. So that's X and an X. Two mutant X's, we'll say. Okay. That's a woman who's got color blindness. And then we've got a mutant X and a normal Y. And so mutant. Okay? So we've only got, in this situation, there's a 50% chance that you'll have a boy that will have color blindness. You'll also have a 50% chance of being a carrier. I mean, 25% chance of being a carrier, 25% chance of being a female with color blindness, and then one normal. Okay. If a carrier female marries an unaffected male, what fraction of their sons will be affected? Okay, so she's got an X and then she's got an X. And here on top we've got an X and a Y. That's a normal male. So we have a normal female and we have a normal male. Then we have an X female who's a carrier, and we have an XY male, okay, who's actually got the disease. So if a carrier female marries an unaffected male, what fraction of their sons will show the disease? We're talking only about sons here, so it's only this group. We don't have to worry about the females. So what fraction of their sons would actually show the disease? And it's 50%. They said, if I asked what fraction of all the children would actually show the disease, then it would be 25%. Okay? So just make sure if I'm asking about one gender or all the kids. So if you are a colorblind male, which parent gave you your colorblindness? Mom, exactly. Okay, so we've been talking about qualitative traits, like single gene, and the expression of which gives you a certain kind of trait. Whether it's flower color, or cystic fibrosis or not, or earlobes attached or not, those kinds of things. But there are also quantitative traits, or quantitative loci. They're called QTLs. And it means that there are many different genes, each with a very small effect, that add up to some larger effect. So a good example is height. We see all different kinds of height here. Okay, and this is height from, let's say, 5 to 7 feet. And for men only, well, actually, we'll say just for women and men. And what in this case, you get a hump near the women, another one near the men. Generally, it's a bimodal distribution, but in either case, there's a whole bunch of different loci that add to how tall you're actually going to be. Skin color is another one. When we look at skin color, there's all shades, right? That's because there are many genes involved in skin color. And skin color and hair color, these kinds of things, have actually evolved independently. There are different genes that are used that express melanin in different ways. I guess musical ability, but there's a certain component, well, it seems to run in families, okay? But at the same time, you often have kids that some are really good and some are okay and whatnot. And then cholesterol levels are another quantitative trait. So quantitative traits actually show continuous variation. Okay? Most of the variation is around what we call the mean. Okay? So you add up each individual's height and then divide by that number of people and you get the mean. Okay? And that's when you see quantitative traits. Qualitative traits, you're more likely they're either or. Incomplete dominance, like with a pink flower, it's not quite, it's actually just one loci. And you get 3 chances. You can be white, pink, or red. Okay? So a single gene that causes a certain trait is called monogenic. Okay, just one loci, and it depends on the alleles that you get. Many genes are called, if this is monogenic, what do you think the other ones are called? Poly, yeah. Okay, yep. Okay. We already know this, which is more likely to show continuous variation. Yeah. Good. So eye color is actually polygenic. There's actually only 3 genes, but you can get with 3 genes and however many alleles, you can get all sorts of color. So anywhere from brown, I think, let's see here, green, amber, and then of course aqua, and then finally blue. Okay, so a famous actor before your time, Paul Newman, had these brilliant blue eyes. Now, as I mentioned before, remember, like, the environment has a huge effect on fitness, right? So some alleles are going to be beneficial in some environments and not. Okay. At the same time, you can have a situation where you have identical alleles, but an environment will cause different expression patterns in terms of how much of a protein, let's say, is made. So this is just someone, obviously, who's been in the sun a lot. But there are often things like, there's a, so most trees, they have branches and leaves and all that. Okay. There's also a kind of form of a tree, not necessarily by species, that is all bent over and has short branches and short leaves. And this one is always found at high elevations where there's lots of high winds. So you end up with a phenotype, even though you end up with the same genes and the same alleles, they're expressed in terms of how they're expressed changes. So we talked about gene expression, and this is exactly what this means. You can have the same genes, same alleles, but different expression patterns. It's actually called phenotypic plasticity. Okay, so it's often seen, like a good example is there are some snails that if they're born in an area where there are not certain predators, these are marine snails, they grow up with kind of a normal swirly curve, and shell thickness is usually kind of thin. But if they grow up in an environment, in other words, they hatch from eggs where there's a predator around, and that predator actually, just by being there, let's say, releases a chemical, it causes those alleles to grow unequally in terms of how thick the shell is and the shape of the shell. Okay? So it has nothing to do with the different alleles, it has to do with how they're expressed in different environments. So as I've kind of been talking about the whole time, you often hear this term nature or nurture. In other words, are we born with these things, or do these traits that we have are they all that way because of the environment we're brought up in? Okay. Well, neither of those things is true. In fact, it's a completely wrong model. It's always genes are necessary but not sufficient. It depends on the environment in which they're expressed. So it's always gene by environment interactions. It is never genes only or environment. Now, in some cases, genes might play a bigger role or a lesser role, and vice versa. But it doesn't mean that the expression, you can still get different expression patterns just because of the environment-gene interaction. Okay, so heritability, as I mentioned, you are half related to your parents. You're half related to your full sibs. You're a quarter related to your half sibs. Quarter related to your aunts and uncles. And 1/8 related to your cousins, first cousins. But sometimes a trait is not fully heritable. Okay, now it's fully heritable if you only have a trait that's associated with one gene and you have different expression patterns based on those alleles. So that trait will be probably fully heritable, depending on which alleles you get. But as we've seen, even in certain genes that are like that, there's still differential fitness depending on the environment that they're in. Okay, so skin color and height are both quantitative, polygenic. They're still about 80 or 90% heritable. That means the loci and the alleles you get are really controlling a lot of the height, but not entirely. And I'll give you an example. Once upon a time, there was this family from North Platte, Nebraska. And this couple had twin boys. They were identical twin boys, right? And the grandparents were so thrilled that they decided to send the couple on a voyage around the world, they'd never been outside of North Platte, Nebraska. So they got on this cruise ship. Now, if you've ever been on a cruise ship, they feed you, well, if you didn't have diabetes before you went on it, you certainly had it afterwards. Okay. So they feed you like crazy. The thing is, some people get a little sick from seasickness, and they have stabilizers and all that, but if it's a really violent storm, it doesn't work so much. So they start out in the Caribbean, things are nice, and then a hurricane hits, and all that food that they ate came up. So already the trip started going badly. And these people are not used to traveling. So they can't wait to get on dry land. They end up in Jamaica. They go off for a walk. They get held up by a panhead ganja smoker, get robbed of everything. It's like, what's going on here? But the worst is yet to come. They're going around the world. So they actually go around the Horn of Africa, or the tip of Africa, and all the way up into the Gulf of Oman, off of Somalia. And what should happen? Piracy. Now, these aren't the kind of pirates that you've been led to believe are kind of cool, you know, with R, matey, and all that kind of stuff. These are pirates with AK-47s and grenade launchers. And they climb on the boat. Now, before they show up, everybody's down at the pool. They have probably about 3 different pools. They're down at the pool. The kids are, you know, peeing in one of the pools. Of course, that's what little babies do, pee in the pools. And so everybody's having a good time. And then in terms of our 2 parents with the identical twins, one of the twins starts getting cranky and needs to take a nap, while the other one, they're just going to let him play with dad down below. So they go upstairs and write, and mom's got a headache, and the kids finally settles down, they go to sleep. Well, that's when the pirate's bored. And they've got AK-47s and stuff, and as they first show up, everybody gets scared, and all of a sudden you see these little brown things blobbing in the pools. So everybody's scared. It's really a mess. But the worst of it is that all the kids go scrambling for their kids, and the pirates kind of see this, and they don't like it. So they, like one of the kids that we're talking about, he gets shot. So his dad's dead. And then this kid is actually captured for the slave market in Chad and Niger. Okay? Now, all in the meantime, they never find the other mother and brother. They go back to the United States. Mom never even mentions this kid actually has an identical twin. The kid in the United States, he grows up, he's beef fed, he's had good diet, he grows up to like 6'4", 250-pound linebacker, right? Being recruited by the University of Nebraska, whatever. Well, the other one ends up in Niger, and he's been bought by a Tuareg tribesman, and that's who's mainly practicing slavery there, and he grows up as a slave. Now, in that environment, what do you think he's eating? He's eating it's shit, right? Probably just like a thin, gruely porridge. Now remember I said that height is like 80% heritable. So he actually grows up to be, let's say, 5'11", maybe 6 feet, but he's skinny. Well, the other brother ends up at 6'4", and really well built. Which one do you think requires more food?
The one that's well built.
Yeah, exactly. Okay. So in an environment like that you're well built, you're great. If you can't keep on the weight, well, it still doesn't hurt you that much. But in the other environment, if you need if you need food, and anybody who's actually smaller overall and doesn't need as much food has an advantage. Now these kids are identical. So one is shorter than the other and less well built the other one. So eventually, the linebacker kid decides he actually wants to go join the Marines, and he joins the Marines. And one of the reasons we got into Iraq long ago was this belief about yellow cake uranium that's used to make weapons grade fuel. Well, they go into Niger, and lo and behold, there's this kind of kid, or he's now a man, and he looks remarkably similar to this Marine. And they're long-lost brothers. So this is an example of phenotypic plasticity. Depending on the environment and how things are actually expressed, you're going to end up with different phenotypes with the same exact alleles. Okay, so even there you can see how environment might play a role. Eventually they bring the slave back to the United States. He actually got married there, and they bring his wife, and they're both their wives are named kind of Marcia, one in Tuareg and one in English, and they come back and they're all happy. The thing is, we get a new president, and everything just goes to hell. And suddenly we're in a famine. Which husband is more likely to survive?
The one that was the slave.
Right, exactly. Okay? So he does. Now, while the other one doesn't. They're identical twins, though. The slave goes on to have, let's say, 4 children, 2 of whom make it to adulthood. So he's relative fitness of 2. Now, the other one died before he had any children, so he had a relative fitness of 0. But remember, they share 100% of their alleles. So what's the fitness of the dead one?
2 as well.
Yeah. In fact, he could have, that one could have been dead at childbirth and still had a fitness of 2 because his other brother survived and produced 2 offspring that then, mated and were successful. So that's something to think about. Okay, here's another one. There are these identical twins, Jack and Oscar. They grew up on Trinidad, but they were separated at birth because mom was the German Catholic and dad was Jewish, and eventually became an American Jew. So Jack grew up in Trinidad in the United States with a Jewish father, and Oscar grew up with a German mother, and he was actually part of the Nazi youth. He was a full-on Nazi, He's actually half Jewish, don't tell anybody. Okay, so there's this YouTube, I'm not going to show it to you, but you might want to look at it. They eventually tracked each other down, a Jew and his Jew-hating brother. Yeah. The meetings at first didn't go well, but eventually they decided to put everything aside and they started meeting. They had a number of surprising characteristics. They both liked, for example, to sneeze to make people jump. It was a fake sneeze, but they'd really go, whoo! Right, something like that, right? And they had a bunch of other things. And remember, they're brought up in much, much different environments. And yet they actually ended up being remarkably similar. So you can see how genetics might play certainly a pretty strong role in such an example. Oh, I know, I have a question. In terms of looking at heritability versus environmental influence, what would tell you the most? Normal siblings, fraternal twins, remember they're about the same as normal siblings, identical twins in the same environment or identical twins in different environments. What would tell you most about heritability and versus environmental influence?
Identical in different environments.
Yeah, exactly. And that's exactly what happened in the mid-70s. A group in University of Minnesota started getting together all these identical twins, some that, didn't grow up in different environments and some that did, and now they've got, a probably a couple thousand twins that grew up in separate environments, and it's been very, very useful. So, for quantitative traits, it's difficult to predict the phenotype of children from the phenotypes of their parents, because you've got so many different genes that are involved to small effect. Obviously, they can have a strong influence on complex behavioral traits. They can also, environment can certainly have a strong influence on behavioral traits too. Think about those two twins. Some things they were doing similarly, and other things they were behaving quite differently. And then the environment, so that's kind of what we've got here.